Hi there, my name is Willow Hoins, and this summer I got to work with Rosemary Carroll and Sam Pierce with PI on creating a study around the effects of oxygenated waters on iron 2 concentrations in beaver impacted floodplains. I don't know what to do that. Oh, sweet. Thank you. So why did I choose this system? Floodplains have disproportionately concentrated hydrological and ecological processes, as well as biogeochemical cycling capabilities. And what that means is it enables them to serve a number of different functions, like depositing fertile soils rich in minerals to farms and ranches, providing biodiverse riparian habitat, dampening peak flows and recharging groundwater, as well as reducing flood impacts. And they're especially adept at sediment, nutrient, and organic matter exchange. But none of this would be possible without the beaver. These guys are the ecological engineers for all of these processes that help create and sustain them. I'm particularly excited about the biogeochemical cycling. And what that is the exchange of ions or compounds across biotic and abiotic components of a system. All of this is made possible by redox reactions. So redox, or reduction oxidation, is a chemical reaction in which oxidation states change, or just a transfer of electrons. So compound A loses electrons and compound B gains them. This also dictates the forms of those compounds and therefore their availability or capability within a system. So redox overall controls the solubility and mobility of nutrients in these environments. And why am I looking at iron? Number one, iron is a highly visible parameter. It's also critical for a lot of biological processes, and most importantly for me, it's redox sensitive. So iron has two distinct species, iron 2 and iron 3. And the ratio between those species helps indicate what the redox conditions in that environment is. So if you're looking at iron 2, you'll see really soluble iron. It's highly mobile, and it can be transported really easily by those biotic materials, especially by water, so really quickly as well. And when you have a high presence of iron 2, that indicates reducing conditions. Iron 3, however, is insoluble. It usually precipitates out into a hydroxide or something like ferrihydrite, which you see here, and it combines or binds on with a bunch of organic matter. That's a lot harder to transport, a lot slower, so that can only happen through erosion. So if you see a lot of iron 3 in the system, it indicates oxidizing or highly oxygenic conditions. So every time that there's oxygen present, iron 2 will precipitate into iron 3. But I want to study what can be transported by the system. So I'm looking into iron 2. And an important part about this relationship is thermodynamically, iron 2 will always precipitate into iron 3 whenever oxygen is present. So it has a really inverse relationship with oxygen. If you see high oxygen levels, you'll have low iron 2 measurements. And if you have low oxygen levels, you'll see high iron 2 measurements. Sam was entering his first year of his PhD, and we got to cover a lot of ground, look at a bunch of different questions, a lot of different variables, clear over 800 samples. But today, I just want to focus on these three main questions. And to study those, we focused on two location sample sites, including Confluence, which looked at the Slate floodplain, which is a sister floodplain to the Pump House, or the East River floodplain. At both of these sites, we took 2 channel location samples, 3 pond location samples, and 3 seep locations. A seep, for those of you who aren't familiar, is just a low flow pooling of effluent water from ponds that has high nutrient contents. So the typical cycle of water in these systems is coming from channels, sourcing into ponds, and then ending up in seeps, which will eventually go back into the river. The variables that I examined were dissolved oxygen, looking at both point and continuous dissolved oxygen measurements. Also iron 2, testing the reliability of 2 different methods, including ferrozine and chemetrics kits, as well as taking in-person and automatic samples. And my final parameters were pH, specific conductivity, and alkalinity, which all just help frame the redox environment. For my first question, is there a correlation between iron 2 concentrations and dissolved oxygen levels in beaver impacted floodplains? Yeah, there totally was. So at Confluence, iron 2 and dissolved oxygen had a really strong inverse relationship. So roughly 63% of the time, 63% of iron 2 variability could be explained by dissolved oxygen variation, which means that dissolved oxygen variation dominates this system's iron availability. However, at Pump House, we maintained an inverse relationship, but it wasn't statistically significant. So roughly only 29% of the time could iron 2 variability be explained by the variations in dissolved oxygen. This indicates that other factors are at play in the system, things like different bedrock chemistry, diverse hydrodynamic conditions, differing microbial communities, their activity and their diversity, as well as competing ions in the system. For my second question, I looked into the relationships changing across ponds, seeps, and channels. And we found that seeps are chemically different. Seeps were significantly different with respect to the variables that we measured. And here you have cluster 2, which is predominantly made up of ponds, cluster 1, which kind of overlaps with 2 and has a mix of the source types, and then cluster 0, which is completely separate and only made up of seeps. This is awesome. We used a Tukey test to determine how different cluster 0 was from 1 and 2, and it was statistically significant. And that's great because it means that the water is significantly altered from the point it entered the system as a channel flow to the point it goes to ponds and then finally ends up as a seep, which means it's been transformed by the biogeochemical cycle. Looking into some of the main correlating variables for these seeps, at cluster 0, which is highlighted in orange, the statistically significant variables included dissolved oxygen and iron for the clustering assignments, which is great because it means that seeps are typically characterized by that inverse relationship of high iron and really low dissolved oxygen. To study this further, at our site, we split it into each of the source types, pond, seep, and channel, against all of our variables, and then ran an ANOVA test to see which ones were statistically significant there. And that, again, was our iron concentrations, our dissolved oxygen, and our alkalinity. So seeps are here characterized by high iron, low DO, and high alkalinity, which makes sense because it means more microbial activity. We saw similar results at Pump House as well. And for our third and final question, is there a diurnal relationship between iron 2 and dissolved oxygen in beaver ponds? So diurnal biogeochemical cycling is really exciting, really interesting, but something really important about redox conditions and biogeochemical cycling overall is asking when those questions or when those measurements are taken. So here we have our preliminary measurements of a mini dot logger tracking dissolved oxygen in our pond over several 24-hour periods. If you just follow the red line here, you can see that most of the measurements are taken during the daytime, which is only during this half of the cycle. So those are only oxic conditions, which means you're only going to see chemical species that can exist in those environments, which means you're missing roughly half of the cycle and maybe half of the species that can all exist in these anoxic periods which happen at night. So severe underrepresentation of the aerobic respiration and redox dynamics overall in studies. And we attempted to look a little bit more into that. So if you saw any tired REU students run around talking about zombievers, that would be our 24-hour sampling campaign at Confluence. We did 2 sampling campaigns. The first measured in-person iron and auto-sampled iron, as well as point and continuous DO. And the second only measured auto-sampled iron and point and continuous DO. So our first sampling was really about fact-finding. Here we had in-person iron measurements versus the auto-sampled iron measurements, point DO versus continuous dissolved oxygen. And what I want you to mostly focus on here is the fact that, like, you know at some point these ponds will go into an anoxic period, right? That's a guarantee in the 24-hour cycle, but you don't know when that's going to be. So at some points that could be at 6 p.m., midnight, or even 6 a.m. And when you're sampling in person, you're only getting feedback to those conditions as you sample. So we may have completely missed some of these cycles. That's helpful. So some of the things that drive this include photosynthesis, wind mixing, and microbial activity, but we wanted to try again for our second attempt, and we only use automatic sampled iron data. So you go into the field, you pull your DO logger, and you can find exact time points of when it's oxic or anoxic, and then pull those samples from those specified hours. Fun times. And it worked. Iron 2 was inversely correlated with dissolved oxygen, as expected, and it was statistically significant with a p-value of 0.014. We also reviewed some additional work, questions, and data collection, including how is iron and DO changing across floodplain types. That's our Trail Creek data. Are the pond's oxic DO fluctuations dominated by photosynthesis? We collected light data for that. And does available carbon impact iron concentrations, or how are other redox elements impacted? For that, we collected total organic carbon data and ICP-MS data. Quick summary of the findings. Iron 2 and DO are inversely correlated in beaver impacted floodplains, but it varies by floodplain. So another finding was that seeps are chemically different than ponds and channels. And finally, that there's a distinct diurnal relationship between dissolved oxygen and iron 2 in beaver ponds, but you have to do really labor-intensive continuous sampling in order to catch that. Some future work includes really exciting directions into different metabolic pathways or microbial interactions. As you can see here, you have iron 2 precipitating into iron 3, and oxygen isn't even present in the system. So other ions, and in this case nitrogen, might help facilitate that role. So it'd be great to quantify that and look into those relationships, as well as sequencing different iron microbes and their interactions with the community. Also, looking at different flow rates and iron concentrations would allow you to do a mass energy balance of the system and see whether it's an iron sink or an iron source, and quantify the system loss. Also, taking a greater breadth of our seeps would be really exciting because we tried to do diverse depths of seeps, but not necessarily diverse activity. Some acknowledgements. I want to thank the Rocky Mountain Biological Laboratory and all my other funding partners for how allowing me to study here and be here. I really want to thank my collaborators and peers for being a great support system, including Sam Pierce, my mentor, Rosemary, my co-mentor, Marty Briggs for loaning his DL loggers, Western and Robin for letting us use their plate reader, John and Abe for braving the 24-hour cycling, and especially John for collecting data points when I blow up my knee, and Susan and Rosemary for facilitating this whole process. I could not have done it without such a great community, and especially without the beavers. So, any questions?
We have time for questions.
Andrew, do you have one?
What are some of the biological or
Like, if they have more iron?
Yeah, like you said those seeds were different by you.
Yeah, so they're basically at the third stage or a final stage of the biogeochemical cycle. So those guys are fulfilling a different role in that cycle. So it's not necessarily that they have more iron or have more ions there, which they do, but it's the fact that they are assisting in fulfilling that cycle. So if the water comes in with a lot of iron that's super soluble, or that water comes in and then it picks up iron or other ions through the system, like in the ponds when you have a lot of concentrated nutrients, then seeps help remediate that back into the system, as well as ponds when they go through these different oxygenation cycles. Any other question? Did I answer your question?
Yeah.
Maybe. Okay. We'll circle back. Ian?
You showed a really cool graph that compared the hand sampling we did overnight to some other body. It looked like those had a similar variable pattern and they were possible. What was responsible for these being automated?
So you're talking about this guy. So when we're looking at this one, if we're taking in-person iron measurements, it means at 3 a.m. we were out there and we pulled that sample and we put it directly into the ferrozine, which preserves the pH and a lot of the other redox conditions that preserve the iron right then and there. But the automatic sampler pulls water at all of those periods, but it doesn't necessarily preserve it in an acid right then and there. So we're getting it in the morning after it's already pulled all of the samples, which might account for some of this dip down. And then as far as for the point dissolved oxygen, we're using a colorimetric kit, which we tested the reliability of it for iron as well. But typically you're handling samples a lot more with a syringe. You might be oxygenating more. These vacuum valves might be getting different volumes of the sample into the vial instead of consistent volume. So that impacts a lot of the measurements, and they were all biased to be higher for the point dissolved oxygen, as well as for the colorimetric iron test that we did as well.
Other questions?
Thanks.